This video is about Bacteriophage Lambda. Bacteriophage Lambda is a virus that infects the bacterium E. coli. When it's not in an E. coli cell, lambda is in the form of a phage particle. The phage particle has a head and a tail which are both made of protein. The head is in the shape of an icosahedron with a diameter of 0.05 micrometers. The tail is tubular with a length of 0.15 micrometers. The phage particle also has a molecule of DNA, which is found in the phage head. It is double-stranded DNA, and when it's in the head it's partially dehydrated, which causes it to take the A form. Here's an electron micrograph of some of the Bacteriophage lambda particles. Here's a drawing of an E. coli cell, with a zoomed-in section of cell wall. E. coli is a gram-negative bacterium, which means that it has two cell membranes, one outside the cell wall and one inside the cell wall. There's a protein in the outer membrane called maltoporin. The cell uses it to transport maltose, but it can also act as a receptor for the tail of the Bacteriophage lambda particle, allowing lambda to adhere to the cell. Lambda can then inject its DNA through the outer membrane and the cell wall. The lambda gets its DNA through the inner membrane using the cell's phosphotransferase system. So the lambda DNA is now inside the E. coli cell. It's now coiled in the B-form double helix because it's not dehydrated anymore, but I've drawn it without the coils to highlight the overhanging 5' ends. These long single-stranded ends prevent host exonucleases from chewing up lambda DNA. These two 5' ends are complementary to each other, and they're GC-rich, which means that they form strong bonds. These factors cause the DNA molecule to circularize. The molecule is then ligated to become a closed circle of double-stranded DNA. But from now on, I'll show the double-stranded DNA as a single line. The circle of DNA is then negatively supercoiled by host-encoded DNA gyrases. This causes the two strands to melt apart at AT-rich regions. These regions are promoters and the origin of replication. The melting apart of the promoters allows the transcription of lambda's genes to begin. From here on in, there are two possible pathways: lysis and lysogeny. The pathway towards lysis begins with replication of the lambda DNA. Then, the proteins for the heads and tails of phage particles and the proteins for lysis are made. The DNA is packaged into the heads and then the tails are added. There are about 100 phage particles in each cell, but I've only drawn 5. There are 3 enzymes involved in lysis. They are all encoded by the lambda DNA, and their functions will be shown in this box. Holin creates holes in the membranes. Endolysin cleaves the cell wall. And another enzyme, the product of the gene RZ, causes the inner and outer membranes to fuse. The cell is now lysed. There are lots of holes through which the phage particles can leave the cell and go on to infect a new host. Now, lysogeny. In lysogeny, instead of being replicated, the lambda DNA becomes incorporated into the host DNA. There is a site on the phage DNA called ATTP, where the P stands for phage, and there is a site on the E. coli chromosome called ATTB, where B stands for bacterium. The lambda-encoded enzyme integrase acts as a catalyst for recombination between these two sites, thereby incorporating the lambda DNA into the host genome. Note that ATTP is not the same site as where the DNA originally ligated when it became circularized, and integration changes the gene order of the phage DNA. When the lambda DNA is within the host chromosome like this, it is known as a prophage. The host cell containing the incorporated phage DNA is known as a lysogen. Now when the host cell replicates, the prophage gets replicated as well, allowing lambda to copy itself without destroying the host cells. In any lysogen, there is a chance of the phage DNA excising from the host DNA and switching to the lytic pathway. Often this is triggered by damage to the host DNA. The prophage senses that mutations are accumulating in the host cell and seizes the opportunity to replicate and get out before the host cell dies or the prophage DNA becomes damaged. In the laboratory, UV light is often used to switch phage from the lysogenic to the lytic pathways.